Hello and good afternoon. My name is Rajiv Nepal. Today I'll be talking about breadth of coverage of MenB-fHbp binding protein vaccine. This is our disclosure slide. All authors are Pfizer employees and they may own company stocks. And today I'll be mitigating any potential bias by only presenting evidence-based content. Encapsulated bacteria have been the cause of bacteremia and meningitis in children and older adults. The polysaccharide capsule, a key virulence factor, have been successfully used as antigens to develop vaccines against encapsulated bacteria. The capsular polysaccharides are generally homogeneous and conserved, so antibodies raised against them cover all strains that share same capsular structure. Now, in case of meningococcal B bacteria, the polysaccharide capsule could not be used as a vaccine antigen because of its structural similarity to human neural tissues. Hence, the use of a surface expressed protein as antigen was the only other option to develop a vaccine against MenB. Unlike polysaccharide capsule, proteins and MenB strains have a high degree of genetic heterogeneity, with each protein containing many variants. For a protein-based MenB vaccine to be broadly protective against circulating MenB strains, the antibodies generated by the vaccine antigen need to protect against most of those variants. The objective of this presentation is to synthesize the existing data on the breadth of coverage of MenB-fHbp vaccine for key stakeholders who are responsible for clinical and public health decision making. As we all know, NACI has recommended MenB-fHbp vaccine both for public health level decision making and individual level decision making. The high diversity of MenB strains paired with the rapidity of invasive meningococcal disease creates several issues. For public health, it may limit the ability to ascertain vaccine coverage of a particular strain in real time during public health emergencies. Similarly, healthcare practitioners vaccinating individuals prophylactically would like to understand the potential coverage that a MenB vaccine would have against circulating MenB strains. In both of those scenarios, a MenB vaccine with coverage against most of the circulating strains would be ideal. So factor S binding protein meets all those basic tenets and has emerged as an ideal meningococcal antigen. So what are the rationale behind the broad coverage of MenB-fHbp as an antigen? As I indicated earlier, based on genetic and immunological basis, the different variants of FHBP can be classified into two distinct subfamilies, namely subfamily A and subfamily B. In its naturally occurring form, factor S binding protein is a lipoprotein, a protein with lipid tail. And there are many studies that have clearly demonstrated that lipid FHBP antigens are more immunogenic than the same variants expressed without the lipidation tail, which result in a broadly cross-reactive responses to variants within the same subfamily but not to the other subfamily. So in order to provide a broad coverage of MenB strains, MenB-fHbp vaccine contains one lipidated FHBP variant from each of the two subfamilies, subfamily A and subfamily B. So let's summarize how we assess the breadth of coverage of MenB-fHbp vaccine before we go into the detail. First step, by using serum bactericidal assay, SBA, we demonstrated that the vaccine-induced antibodies were able to kill different variants of FHBP from both subfamily A and subfamily B. Vaccine-induced antibodies mediate the killing by binding to the FHBP expressed on the bacterial surface. Hence, in the second step, by using MEASURE assay, which is a meningococcal antigen surface expression assay, we establish the threshold of FHBP expression in a bacteria that is essential for the killing in SBA. And in the final step, we determined what proportion of Canadian strains express FHBP above the threshold. This helps to estimate the breadth of coverage of the vaccine and circumvents the need for doing SBA against every single strain, which is neither practical nor feasible. Now let's discuss each of these steps in detail. The serum bactericidal assay measures the killing of meningococci in the presence of specific antibodies that bind to the bacteria and activate complement cascade. To perform the SBA, MenB-fHbp vaccinated subject serum, which contains antibodies, is mixed with MenB bacterial strain under test. If the MenB is killed, the vaccine is considered to have coverage against the particular strain. In this slide, by using SBA, we show how MenB-fHbp vaccine can kill diverse strains carrying FHBP variants from either subfamily A or B. The two subfamilies of FHBP are represented here as two separate flowers. The MenB-fHbp vaccine, as I said earlier, contains one lipidated FHBP variant from each of the two subfamilies. So those are A05 and B01 and are represented in the center of each flower. The approach to evaluate the breadth of coverage started with SBA assays showing the killing of 4 primary strains, 2 with subfamily A and 2 with subfamily B. To further assess the breadth of coverage and to support the use of these 4 primary strains as representative of broad coverage, 10 additional test strains from both subfamilies were tested and shown to be killed in SBA. All these 14 strains were vaccine heterologous and represent endemic and outbreak strains from the US and Europe and were also used in the pivotal phase 3 studies. There are a number of additional studies, both internal and external. They have used sera from subjects vaccinated with MenB-fHbp to demonstrate the killing of diverse meningococcal strains, many different strains which have collectively expressed 33 FHBP variants, 18 from subfamily A and 15 from subfamily B. Interestingly, 9 out of 10 prevalent Canadian FHBP variants from MenB strains that have caused invasive diseases here in Canada, they have already been tested in SBA and they are found to be susceptible to MenB-fHbp induced vaccines, and those strains are indicated by the flag on the flower. Now let's discuss the second step. Since antibody cross-linking is important for complement deposition and bacterial killing, sufficient FHBP expression levels are required for killing in SBA. An isolate with very low FHBP expression may not be susceptible to killing in SBA. Hence, we sought to establish a predictive threshold where most strains were susceptible in SBA. And measure assay was used to measure the FHBP expression of those different strains. On the graph to the right, the x-axis represents different MenB isolates and the y-axis represents the expression level on that particular strain. The colors denote whether a strain was killed in SBA, with green diamonds representing killed isolates and red triangles representing isolates that were not killed. The dotted line there defines the threshold where the majority of strains were found to be susceptible. In the third and final step, Beringer et al. In their 2020 publication measured the expression of FHBP by using measure assay among Canadian meningococcal B isolates, which were collected as a part of the impact surveillance between 2010 and 2012. And 91% of the isolates expressed FHBP at levels above the threshold that I just described earlier, and they are predicted to be killed. So in conclusion, and as a take-home message, 9 out of 10 Canadian MenB strains that caused invasive disease here in Canada were predicted to be covered by antibodies generated by the MenB-fHbp vaccine. So here is a one-pager infographic that we all are working on, where we have condensed the information. And with this, I would like to conclude my presentation, and I'll stick around and I'll be happy to answer if there are any questions at the end of the session. Thank you very much for your attention.